Thank you so much. I'm Vanita Dahia. I am a pharmacist, naturopath, nutritionist. I've completed my fellowship in anti-aging medicine, both in the US as well as in Australia. I've done my diploma in Ayurvedic medicine, so I've dabbled in a few modalities and realized that each modality has its limitations. Now, when we're talking about chronic inflammatory conditions, and I don't care what brand name you like to attach to it, whether it's chronic fatigue syndrome or whether it's MND or MS or SLE, they all have immunological issues that are going on. And we need to rectify those immunological constraints. And the issue now is that when we go to the doctor, as Peter had mentioned, we only hear about the normal ANAs and ESRs and your CRPs. That's on, yes, thanks. And that's very, very important to get an idea of some of the assessment options that might be available. Because when you're feeling sick, a lot of the time doctors would say to you, well, my dear, I think it's your fatigue or your pain is all in your head. Go home or go and have an antidepressant. And often that might actually work for some cases because there could be a neurological association, but we now need to know that there are other underlying causes. There may be infectious causes, we've got to be able to learn to prioritize what exactly we are looking at. Now, chronic complex inflammatory conditions is basically involving the immune system. It affects every organ system. It doesn't matter whether it's the hair, skin, nails, bones. And there may be underlying causes that are not only associated with mitochondrial dysfunction, there could be infectious conditions associated. We now know that PANS and PANDAS, which is a psychiatric disorder in kids, is associated with a confirmed streptococcus infection. All right? So there's enough study now to show that microbes are playing a major role. And with the study of microbial analysis now, we now are able to actually identify these imbalances right up front so that we can start correcting them. So we're going to be talking about all of those options. The types of chronic illness is huge. It's endless. The problem with it all is it's often a lifelong condition. Most often it can be reversible, but it's generally a degenerative condition, okay? So it affects the immune system, and then you can also have acute conditions as well that might initiate and be a trigger for an autoimmune condition. For example, you could have appendicitis or acute leukemia or strep throat, and that can be an initiator of another autoimmune type condition. Because often you'll find some people will say, look, I was perfectly fine, but then I got exposed to my friend's child who had chickenpox. And I had a bit of a cold sore, and ever since that moment I went downhill. So that triggered an immune response and a viral load, which initiated a cytokine storm, and that was your inflammatory response. And that was the initiation. That's the reason when I talk to clients about inflammatory conditions, when did it start? And there's usually a trigger. The types of chronic illnesses are huge. They're thyroiditis, diabetes type 1, Lyme's disease. There's a lot of new work being done on mold, Lyme, biotoxins, mycotoxins, gliotoxins that are now associated with tick-borne illnesses, mold illnesses. Neuromuscular, neurodegenerative conditions. So here you're looking at MS, MND, SLE, they all associated with pain and inflammation. Sarcoidosis as well, with pain and inflammation. Sjogren's is dryness of the body. IBD is very common. IBS, everybody seems to have it these days, which is really fluctuations of constipation, diarrhea, and bloating. But IBD is Crohn's disease. It's ulcerative colitis. So these are the issues that we are now seeing. And then obviously the new events of your pandas, Alzheimer's disease with Cunningham profiling, all of these are the appropriate, I guess, the newer protocols that are available. Now, symptoms are obviously varied. Everybody has different symptoms. Some people talk about fatigue. Others talk about pain. They could have enlarged lymph nodes or sore throats and strep throats or flu-like low-grade infections. That's typical of a chronic inflammatory conditions. Now, could it be associated with diet? We find that amending diet plays a major role in the outcome and the progression of a disease. We know that sugar is a major inflammatory component to your diet. Anything that is processed tends to be that. And the strange thing is that our diet is so heavily laced with pesticides, with herbicides, with adulterants, with colors and preservatives, and now we know that. We're all aware of it. So as a result of that, we have now come across new diets, which are your GAPS and FODMAP dieting and your carbohydrate control diets. These are all now becoming so-called fashionable. And they have really great value for some people and no value for others. Some people who may start a GAPS diet might get really sick initially. And that's because they've never had the diet ever, and they're changing their diets accordingly. But our focus in this session is all about what we can do about testing. The types of infections that seem to affect us are associated with either methicillin-resistant strep or staph infections, Staph aureus infections. That's very, very typical. But we can also get infections that are associated with Pseudomonas, and then we've got, viruses, norovirus infections, whether it's CMV, varicella, or Epstein-Barr, and then we've got your blood-borne pathogens that are also playing a role in infectious, underlying infectious control of your inflammatory condition, whatever it is. Now, when you go to the doctor will say to you, well, let's have a look at some basic FBEs, full blood exam, or if you're in America, we call it CBC, which is complete blood counts. So we'll look at C-reactive protein or high-sensitive C-reactive protein, homocysteine, ferritin. These are the obvious ones that are covered through the Medicare system. They also look at the total levels of IgG, IgE antibodies in the body. They'll also have a look at things like ANAs, and if you really ask the doctors, they might have a look at RFs if you've got a history of osteoarthritis. They might identify if you've got rheumatoid arthritis. But a lot of the time, they don't look at your double-stranded DNA or the smooth muscle antibodies, and this is very important when you've got muscle or myalgia, pain in the muscle. So it's really important to know that even though that ANA is commonly used as a marker of assessment, one should think about SMA as well as the anti-mitochondrial antibodies as well. Like, is the mitochondria, which is the power source of every cell, is it actually making energy, which is ATP? Erythropoietin viral loads, the standard viral loads are usually EBV, CMV, which is your, CMV is cyclomegalovirus, and your varicella. HbA1c is a pre-diabetic marker, and then your nutrients. They might look at your vitamin D, your B6, B12, and your full blood exams will normally consist of lipids, iron studies, and hormones and thyroid. So the doctors would look at this and they'll say to you, well, look, I think it's all in your head. Go to a psychiatrist. It might be, go and have an antidepressant because we can't do anything more for you. So the other assessment options that are not lab associated are things like nerve conduction, where they actually apply electrodes to the nerves, electromyographies, magnetic resonance therapy. These are all your other assessment, nerve conduction tests. transcranial magnetic stimulation, and this is applying your various electrodes to the skull, and so forth and so on. Then there might be, for those Sjogren patients, you might have the scintigraphies, which is measuring your radioactive isotoping in terms of saliva output. In addition to that, now, sorry, that was meant to be lab. These are some of the lab things that we might want to consider. And these are things like a microbiome mapping. Now, gut testing has progressed so much, and I'll introduce you very briefly to the new concepts in gut testing. Diet, obviously, you can identify allergy, sensitivity, intolerance. You can identify always, we've got to rectify the endocrine system. Because a lot of these imbalances stem from possibly an underlying major stress event. How many people have a chronic condition because it started as a result of a major stress event? Okay? It could have been a trauma, it could have been major, something major. Now the body is very used to being traumatized. It can go to a torture chamber and be traumatized. And you will fire at all cylinders to rectify whatever you need to do, and cortisol will always balance and react to that stress at the expense of other organ systems, at the expense of the endocrine system. And so as a result of that, we need to make sure that the endocrine system is optimized. So the adrenals, the thyroid, and the hormones, they all play a role together. If your adrenals are mucked up, then obviously you're going to feel tired. But then that is the precursor to anxiety and depression and a whole bunch of other neurological conditions. It is also a precursor to inflammatory conditions. Then we look at things like cytokines. Now you ask a doctor, can you measure my cytokines? And they'll say, what's that? You know? So we need to be aware that you can actually measure that. There's nutrient imbalances going right much deeper, not your B group vitamins and glutathione. We want to know exactly how is my body producing energy? Where are the metabolic blocks in producing energy? Amino acids, vitamins, and so forth. Genetic SNPs. Genetics have progressed so much now that we have an accessibility of all these various gene SNPs that are available to identify specific ones. I'll bring to your attention just one or two. And every person that is chronically fatigued, chronic inflammation, they cannot work. They have financial issues. They have all sorts of other issues. They can't even get out of the bed in the morning. Okay? Everybody else is having a normal life. And these are people that cannot function normally. So what do we do? Right? So it affects the nervous system. Sorry, just went backwards. Wait a minute. I pressed the wrong one. So basically what we need to do then is to actually start rectifying all of these constructs that affect the nervous system as well. So you can actually measure neurotransmitters as well. So where do you start? So in this session, I'm going to give you a sort of like an overview of options. Now, all these options don't apply to everyone. Everyone is individual. Everyone needs to be focused on exactly what they need. And so you need your appropriate professional advice to give you the appropriate guidance. But now we're going to just give you an overview of all the options that are currently available. Obviously, there are some that are also available that are not in the presentation, but for the context of this talk, I think it's important for you to know what else is available in terms of assessment options. So diet, we'll talk about that. SIBO, microbiome, bowel movements, lab testing, we'll talk about some of these here. Okay, so this is what a typical stool test would look like. This is a brand new test which is called a complete microbiome mapping test. So it's looking at the DNA of bugs and identifying. So it's a lot more sensitive and specific. So in a test result, you might get issues associated with, like, you know, the stool, the form, color, whether there's any blood detected. Markers, this is one test, right? This is called complete microbiome mapping. So calprotectin identifies and differentiates between IBS and irritable bowel disease, which is Crohn's and ulcerative colitis. Pancreatic elastase gives you an indication whether you are digesting well or not. Do you need extra digestive support? Secretory IgA tells you where there's inflammation. Why is it that you're feeling bloated? Or why is it that, remember, 90% of your serotonin and GABA is actually made in the gut. So if you're feeling, if your gut's not in the right space, then this is not going to be in the right space either. Zonulin is a marker of leaky gut. So I'm sure you've heard of the term leaky gut. You know, you're taking all these supplements and you're not feeling any better. Is that, you know, do you have a leaky gut? So leaky gut means leaky brain as well. So you feel, you just can't think straight, okay? So zonulin is a marker of identifying leaky gut. Beta-glucuronidase is a marker of liver function. And then in the report you'll find out whether you've got parasites, bacteria, fungi, and appropriate infections there, and also the ratio of the good bugs and the bad bugs. The good bugs are your commensal microbes. So they divided into categories, the Bacteroides and Firmicutes, and it is well known in the literature when Firmicutes is high relative to Bacteroides, it is associated with neurological disturbances. So your gut bugs, the balance of the gut bugs are telling us what our moods might be like. So just breaking that test down in a little bit more detail, we've spoken about calprotectin. It's a gold standard for IBD. The next one we talk about is your microbiome summary. So it gives you an indication of where the bugs are, whether you've got a parasitic infestation and worms, bacterial and viral infestations, as well as fungal infestations, okay? Then the phylum microbiota, right, when we actually had a look at these ones here, the Bacteroides and the Firmicutes, these two phyla represent about 95% of the good bugs. So when you're taking kefir yogurt or your quark or your probiotics, you are actually consuming the good bugs. We get the good bugs from our diet anyway. Okay, the classes of these bacteria are called actinobacteria, proteobacteria, and they're all gram-negative bacteria. These are your Bacteroides, and Firmicutes are the gram-positive bacteria, and that balance is very important. So people who have an imbalance of this area will have inflammation in their gut, so they might need prebiotic support to rectify that particular pathway. They've actually done, D'Souza and his team have done the study using 30 grams of glutamine has actually helped that area. We've got 4 minutes before you have to switch off. So the parasites and the worms, that's another one. So like in this particular instance, parasites are measured and blastocystis. Now blastocystis is one of those really bad, nasty bugs. Very hard to treat. There is no prescription drug that is used as a single line treatment. There's some beautiful herbs that can be used very effectively. And so it's a parasitic organism which is identified by the polymerase chain reaction technology. Worms as well. So these are your thread worms, your ringworms, all of those worms. And they are also identified. In addition to that, we've got opportunistic bacteria that are measured, and these are organisms that are illustrated the top range. If it's above the range, then this patient has Pseudomonas as well as Staphylococcus infections. And as far as these are bugs that are associated with autoimmune issues. So there are bacteria that tend towards initiating and aggravating and augmenting inflammatory conditions. And those bacteria are things like all of these ones listed. In this case, this patient's got a Klebsiella infestation. So we're not talking about treatment, we're just talking about options of assessments at the moment. Opportunistic bacteria also measured, so these are your lipopolysaccharide that stimulates that inflammatory responses, and they thrive. So things like Klebsiella is a very good inflammatory bug in the body. They thrive on inflammation. Fungi, this patient example has a geotrichum infestation, so you've got to use antifungal strategies and start correcting that. So the reason I'm bringing this test to you is because it's so comprehensive. You can look at one stool sample and it's giving us all of these issues, these levels of assessments. And then viruses as well. Everybody says that's in a chronic inflammatory condition will often say, I feel like I have a low-grade flu all the time. It doesn't go away. And they may have a viral load that is not identified or that may be identified. So we need to address that and correct that as well. And then you're also looking at your good bugs and the bad bugs. So the good bugs are all of these ones here. These are your Bacteroides and then the Firmicutes and then the ratio of them. So if there's insufficient amount of the good bugs, then we need to put them back into the system to drive them up. Sometimes if they're high enough, then that's great. These are the beneficial Clostridium species. If you've got there are many species of the same bug. If you've got Clostridium reported in the opportunistic bacteria as being high, then you would want to treat that as an infection. Whereas some of these good bugs like E. coli, for example, can be good or bad depending on the type of species and genera they come from. Okay? So this is all part of mycology. So who's heard of the term SIBO? Right? Okay. Stands for small intestinal bacterial overgrowth. Now there's a lot more information available on gut imbalances because the seat of disease starts in what we stick down our throats and what happens in our gut. So because if you've got a leaky gut, no matter what good nutrients and pharmaceuticals and supplements you're taking, it's just going to go right through you, okay? Or it's going to create toxicity in the body and it's not going to go to the target tissue that it's meant to. Or if you have imbalances in the microbiota, you're going to have neurological conditions. You'll also have inflammatory conditions, which is the diseases that you have, whatever brand name you have of it today. Now, when you're doing SIBO testing, you're actually breathing. It's a breath test. So you're breathing into a bag and we're measuring the breath following the intake of certain ingredients in the kit. So what we're measuring is hydrogen and methane and carbon dioxide. And the way to very briefly read this, we won't go into much detail, but all I want to let you know that if we see a expected hydrogen at the start at 12 minutes, around here, at around 90 minutes is when we have, this part here is the small intestine. Once it moves from 120 minutes onwards, this is the travel of your hydrogen and methane through the large intestine. So a lot of these imbalances happen at the small intestinal level. So if you've got hydrogen elevated, greater than 20 parts per million, from 12 over the 90-minute period to 57, so 57 minus 12 will give us 45. And that tells us that this patient has an elevation in hydrogen which is greater than 20, therefore this patient has SIBO associated with hydrogen-emitting gases. Conversely, we have methane measured as well. Methane anywhere along the line, 8 being the highest, 2 being the smallest, so the result therefore is 8 being the highest. It's less than 10, therefore this patient does not have a methane-associated SIBO. It's a hydrogen-associated SIBO. Now we won't go into the fine aspects of it all. Carbon dioxide is obviously measured mainly as a quality assurance, but the most important thing here is that your SIBO test will give you an indication of the level of dysbiosis in the small intestine. Patients who have indigestion, patients who have typical patients who have bloating, indigestion, pain in the gut, gastric reflux, belching, burping, bad breath, halitosis, often reactive to prebiotic treatments. They get worse when they're taking prebiotics. They are likely to have SIBO, and this is where this test would come into play. So it's actually measuring what's happening in the small as well as in the large intestine. So what do you do about it? This is all about assessment, so we're not talking about treatments, but the treatments are very, very defined. It involves using a biphasic diet. It involves using a GAPS diet or a FODMAPs diet or an anti-inflammatories support. And there's some really good sources of information out there. There is a SIBOdiet.com website, which is worth you looking into if that is what you're interested in. Then we go backstream and we'll say, well, what exactly are you eating? What's happening for you? You know, you find that when you eat certain foods, this is what's happening for you. You might feel worse, you might feel tired, you might feel... Now, I just want you to be aware that when you go to your doctors and you ask for an allergy test, they do what we call a Rast test. In other words, they prick the finger, the arm, the inner arm, and they potentize it or they activate it or sensitize it with certain allergens. Now, anything is a nutrient if it is absorbed, but once it elicits a histaminic response, in other words, your macrophages say, oh, not good for me, this is an allergen, in other words, it's a baddie, it's not a goodie, then that allergen elicits an immune response. Now, there are 5 types of immunoglobulins in our body. Normally, secretory IgA is like the soldier in the gut. It goes around in the gut and it says, oh, that bread's no good for me. I'm going to scavenge it and I'm going to excrete it out. But the brain still says, look, bread is fine. I'll just keep eating the bread. Secretory IgA in the gut dies down. So guess what? The tight junctions in the gut start to open up. Your bread is absorbed into the body. The body now, the bloodstream says, oh, this is an allergen. This is not a nutrient for my body. It's going to elicit a histaminic response. The macrophages start to swell up, and then you have immune responses. So there are 3 things that we can test for. One is allergy. Now, allergy could be a bit of rhinitis, sinusitis, or the worst case could be anaphylaxis. You have to go and get an EpiPen injection, right? Or you die. That's an IgE-mediated response. An IgA-mediated response, secretory IgA lives in the gut. So nasal mucosa, the respiratory tract, the gut, all associated with IgA. So a sensitivity to those foods is IgA. The next thing is an intolerance. You're eating bread, and you find, oh, look, that's fine. But the next day you feel all bloated and tired, okay? So that is a delayed response. That's IgG. So that's what we're measuring. So when you're looking at an immuno-IgE, this is an IgG. So it measures 96 foods, typically. And so it looks at dairy, and it differentiates between casein and whey, because there's certain dairy products that are casein-rich and others are whey-rich. Your meat products, your fish products, your fruits and miscellaneous things, and then the grains as well, and it differentiates between gluten and gliadin, as well as your vegetables. So that's the sort of test that you can get, and that's just a finger prick. You can also measure IgE, IgA, and you can also measure your chemicals. You can measure your food additives. Now look, I mean, you know, when you buy your piece of meat, you think you're buying a lovely organic piece of meat. It's nice and rich and red in the butcher shop. And if you kill an animal, you cut the meat, it's going to turn brown or gray pretty soon, right? So how does it look nice and fresh and red? They add sodium sulfite to it. You know, MSG, every packet of ready-made noodles contains MSG. You think it's only Chinese takeaway? No, it's in all processed material. So there's a lot of these things that are found everywhere in our food. Your food additives as well can be measured. Your inhalants and molds can be measured as well. Molds can be measured as well, because that's a simple blood test as well. So this can also be, there's a different version of testing, and it's called Alka testing, which identifies the rate at which those cells, remember I said your macrophage, your white cells enlarge, they become inflamed, and so this is what is called an Alka test. So it identifies things like cow's milk colors and herbs and antibiotics, and this is typical. So it'll tell you whether you've got candida, whether you've got gluten intolerance, whether cow's milk or goat's milk is good or bad for you. Good stuff, bad stuff, and really ugly stuff, okay? Question? Yes, it can.
So are you saying you should do that test to see if you're suffering from celiac or gluten, or just gluten intolerance, not celiac?
Gluten intolerance. Celiac is a different blood test, yes. You've got to measure your celiac, gluten and gliadin, IgA antibodies, okay? So that confirms that. So when you're looking at your labeling, it's really important to, this is a really good website, foodstandards.gov. It really gives you an indication of what's cooking, you know, I'm not going to discuss this in much detail, but there's so much crap in our food. You know, there's so many additives in our food. So I tell practically every one of my patients, if 90% of your food consists of fruit, 3-4 pieces of fruit, vegetables, a pile this big of vegetables, whether it's baked or juiced or souped or stir-fried, a pile of veggies. And this is, we've missed the boat. And we think only vegetables are the ones in the supermarket. There's a million vegetables that our grandparents ate that we're not even using today. You know, 3 tablespoons of oil. The brain is fat. We need the oil. Inflammation is a lack of the good oils, and we've moved away from the oils. And I'm not talking about your vegetable canolas and so forth. I'm talking about your essential fatty acids. So these are olive oil, coconut oil, MTC oil, hemp seed oil, flax oil. Put them into your diet. They're very, very important. And water. Most of us are dehydrated. We drink teas and coffees and wines, etc., but we don't drink water. Now, if that's 90% of your food, then you're on the right track. You've obviously got to put the grains in. But we only think about grains as wheat. There are millions of grains that are out there that our grandparents ate that are far richer. There's certain types of millet that have very high levels of iron. And we don't, the problem is we get stuff that is so adulterated. So, you know, I mean, even you buy a packet of oats, it's got antifungals in it because it's in a plastic and it's going to become moldy. So it's got antifungals in it. It's got dehydrating agents, so it looks all nice when you have a nice, quick cook oats. Okay, that's simple. You think oats is like simple, right? And yet it gets it's adulterated. So keep your diet as good as possible. So you've got to know, you know, your numbers, and I mean, this is not a talk about labeling, but it's just an awareness. It's an awareness of what is it that we're sticking down our throats, okay? You know, and you've got to also watch for your allergens, and there's a lot of things like textured soy protein, cornmeal, you know, and so forth. There's a whole lot of junk in our foods. Now that we think about gut, let's say we've got the gut optimized, because that's a really good starting point. We've got to optimize the way the gut is functioning. Increase absorption, elimination, excretion, metabolism, put the right foods in our gut, etc. But we still have this chronic condition. We may have optimized the gut, but we're still feeling like crap. We still have pain, we still have neuropathic pain, we still are extremely exhausted. So what's caused it? Is it an issue associated with adrenal dysfunction, or is there neurological imbalances, or is it the energy that's so damn depleted? So we need to figure that out, okay? So let's have a look at some of these. This is a simple urine test that identifies cortisol. So cortisol is that lovely little stress hormone that is released from the adrenals, little glands that sit on top of the kidneys. So you're writing exams, cortisol goes up, you finish your exams, cortisol goes down, party time, right? But the problem with all chronic inflammatory conditions is cortisol remains up. You're just firing on all cylinders all the time. And you've done that for that long that the adrenals stop functioning. Those cortisol receptors stop functioning. cortisol eventually drives down together with DHEA. And when cortisol and DHEA are down on the floor, that's what we call chronic fatigue syndrome. That's what we call fibromyalgia. That's what we call autoimmune conditions. So that's what we need to do, is we need to look at the stage of adrenal dysfunction and correct it. Sometimes it might need herbs. Sometimes it might need a prescription for DHEA. Sometimes it might need you sitting under a teepee and meditating all day. But we don't get the balance of all those constructs right. So that's what we need to do. So in a urine test, we can measure your cortisol together with cortisone. And generally, you would need to see it in that green line. And obviously, here you can see in the morning, early afternoon, is all high, even late at night. So this person is obviously not sleeping well. They're wired and tired there. They're so depleted at the end, and then they get this late wind at night. Okay? So this is somebody that's at early stages of adrenal dysfunction. So when you actually look at the reporting itself, this is a urine test. When you look at the reporting, they take this information, and when you get your result, it'll look a little bit like this, where you will see that those levels of cortisol are matched against various symptoms. So it's quite typical to see, some of these symptoms that are associated severely. You will typically see that in association with elevations in cortisol. And then at the bottom of that sheet, you will also see what is happening for this patient, the propensity based on those results. There's a propensity for all these conditions based on on their levels of hormones, okay? So that's the degree of information that you can get. See how different it is from those standard numbers you get from your doctors? You can get so much more information. You have a question? Alopecia and fibromyalgia, there's obviously an adrenal dysfunction there. So adrenals need to be identified and rectified. And then alopecia is also associated with testosterone to DHT conversion. So there may be an issue there. And there's also associations with thyroid. So you've got to examine all of that, optimize that, and then alopecia does not equal fibromyalgia. All fibromyalgia patients do not have alopecia. So there's an underlying trigger.
And what about rosacea?
Acne rosacea is usually associated with peripheral vasodilatation. Rosacea could be associated with hormonal dysregulation, as well as, so there's some studies showing that testosterone depletion is associated with rosacea. There are vasodilatory stimulants such as capsaicin and various other green teas, etc., that can also be used. Yeah. So if you're doing a spit test, you can also look at your cortisols through spit. So in this particular instance, this person at stage 3 adrenal exhaustion. So you might find that your cortisol might be low. It should be in the middle. And this person's like really, really low, flatline low. This is a person that probably can't get out of bed. And then, but their DHEA is normal, but their total cortisol is low. So that person's not that bad yet, but if DHEA was really low, that would be a typically chronically fatigued patient, okay? Then there's other areas that we may want to look at. Question? Yeah, so spit test and urine test are pretty much the same. The only problem is a lot of people can't urinate 4 times at the exact time, and this is why spit test is so, for ease of collection, that's better. Okay. Now, I'm sure you've heard of the term pyrols. Have you heard of pyrols or cryptopyrols or mofactor? There are some nods. And so pyrols are basically the neurotoxic compound that comes out into the urine when it sucks up. These pyrrole molecules, they suck up and they mop up all the vitamin B6 and the zinc. Now, Dr. Carl Pfeiffer has done, in the Pfeiffer Institute of Neurological Sciences, has done a lot of study, and now his work is now superseded with Dr. Bill Walsh's work on the nutrient power of nutrients in mental health. And he's talked about lack of certain nutrients like B6, zinc, and omega-6 oils that are contributing towards the severity and the exacerbation of neurological conditions. A lot of the study has been associated with schizophrenia. Putting back those nutrients, B6 and zinc and so forth, at a specified rate, published studies, numerous published studies, schizophrenia alleviation. So this, I mean, you know, we don't need to go through all these drugs that are out there if we can identify the underlying cause and correct that, all right? So that's pyrols. We can also send a urine sample off to the lab and we can have a look at your nervous system. Because every chronically inflamed patient will have a nervous system issue. They will say, I'm feeling depressed, I'm feeling sad, I have suicidal ideation, or I have panic, I feel like I'm having a heart attack. And each of these areas can be identified. So you can have your inhibitory pathways without the inhibitory neurotransmitters. We all know about serotonin, right? Because some of us take SSRIs, they're serotonin inhibitors. As reuptake inhibitors. GABA is another one. GABA is the stuff that makes us get to sleep and relax and calm down. Now we need those inhibitory ones so that we can relax. But we also need the excitatory ones to get us up and going and go to work and get motivated about life, okay? And these are the excitatory ones. So I won't go into the detail of your neurotransmitter profile because I've actually written a book on this called Alchemy of the Mind. But the importance of, when you go to your doctor, you will get a DSM questionnaire and they will classify you based on your symptomology and questioning as to what label they can attach to your neurological condition. This test does not label a condition. It gives you the balance of the inhibitory and excitatory neurotransmitters and gives us the appropriate associations. For example, low GABA, low serotonin and that is based on optimal ranges. So serotonin is 73, and ideally you want it between 200 and 415. So this person's sad, got the SADs, okay? They are depressed. Their GABA is 271, should be around 600 to that. So they are anxious, they're panicky. Now before you do anything else, it is very important to rectify the inhibitory pathways first. And then you start working with excitatory pathways. With what? Yes, so you can use, if you're not using pharmaceuticals, the GABA precursors are things like lactium, glutamine, GABA, L-theanine. Your serotonin precursors are 5-hydroxytryptophan, tryptophan, L-tryptophan, SAMe, B6. Remember, none of this works without the cofactors. Your cofactors are so important. They are B6. In the case of serotonin, B6, iron, tetrahydrobiopterin, which is folic acid. If you don't have those things, no amount of SAMe and tryptophan will work. You need those cofactors to make sure that that will be sufficient to manufacture. What you're doing is you're topping up the tank. Because SSRIs will inhibit, when you're sad, your serotonin from these little vesicles, they get reuptaken when you're sad. When you've got an SSRI coming in, like a Prozac or whatever, sertraline, it inhibits that reuptake so that serotonin that's in the vesicle is now released. And that's why most people who take SSRIs feel wonderful in the first few months. Afterwards, they feel pretty much worse. They feel like a zombie. And the reason is that we're not topping up the actual serotonin in the vesicles. And that's why you need to put those cofactors back in to make more serotonin. All right?
I know there's not a hell of a lot of research into nootropics, but do you know much about them in relation to this?
Which specific nootropics are you using?
Racetams and that sort of stuff.
All your pyracetams, racetams, your ergoalkaloids, these are what we call the smart drugs, and they're used in cognition decline. They're used in, and vinpocetine is another one, and they are used in cognition decline. That's been heavily studied in Dale Bredesen's work on Alzheimer's. and it is available, but it's compounded in Australia. It's not available commercially. It's compounded, particularly in neurodegenerative conditions like Alzheimer's, with Parkinson's disease, with cognition decline, with senile dementia. Neurotropics are really useful for that purpose. When you're using it for a purpose of correcting mood disturbances, they would be secondary. All right, so I mean, I can talk about this area endlessly because this is an area of my specialty. But let's move on because we need to get an understanding of all the options of assessment. Now, I'm sure you've heard of histamine. Has anybody heard of the term histamine? We use antihistamines when we've got a bit of a cough or a cold, or when we go in, when we have the hay fever season, we use antihistamines, right? That's a H1 antagonist. We use Tagamet or cimetidine when we've got a bit of an ulcer. Now it's superseded with your PPIs, like Somac and omeprazoles and so forth. But the traditional cimetidines are H2 antagonists. H3 receptors are found in the brain. So when histamine is elevated, we have problems with methylation. When histamine is elevated, we have depression, we have all sorts of conditions. And so there's a typical scenario associated with histamine. And you need to make sure that if a histamine is elevated, the patient's on a low histidine diet, particularly leftover food, deli foods, those sorts of things are really high in histidine. You've got to make sure that the methylating cofactors are back there. And then diamine oxidase, histamine does two things. It gets broken down by HNMT, which is histamine N-methyltransferase. That's a methylation process, as well as diamine oxidase. Now there's a lot more study on diamine oxidase. This is copper-driven. And this is why nutrients such as copper and zinc play a role in mental health. Okay? So I know that from a lay person's perspective, it sounds like a lot of gobbledygook, but it all makes a lot of sense when we look at the biochemistries. Biochemistry is wonderful. It's a beautiful picture. You don't even have to know anything about the patient. When you understand the biochemistry, it reveals all these metabolic blocks, and then you could start fixing them.
Sorry, what test is this one?
That's a histamine and diamine oxidase test.
With histamine,
Yes.
if we've got SIRS, should you avoid histamines like the foods?
It is very, very important that SIRS, you don't avoid, there's no blanket diet for SIRS. You know, they use a low oxalate diet for SIRS patients. And you can go and have a look at what's, you know, what's low. There is Dr. Richie Schumacher diet under the SIRS. There's a really great diet there. And yes, there's certain instances where histamine is not appropriate. The GAPS diet might not be appropriate for certain conditions. But you cannot say all SIRS patients should avoid histamine-containing foods.
So I
Okay?
used to make my kombucha and
Yes.
ginger beer, and I'll cut out kombucha.
Yes.
But I just have fermented ginger beer. But I am on a vegan diet, so
Sure.
that helps.
Yeah, yeah.
Because I know,
You know?
like the bulletproof diet has things like avoid mushrooms with SIRS, you know, avoid
Yes.
corn, and you're already gluten-free, so it's sort of like
So it limits the amount of
gluten-free.
food that you can eat. Yes,
Yeah.
yes. Another pearl, there are very, very many more diets. There's never been a time where there are many, many more diets and more evidence to show that this diet is better than that diet, and this study has shown that that diet's better than that one, and so forth and so on. So I'm not subscribing to a specific diet. What I'm subscribing is go back to your ethnic diet. What did your grandparents eat? Because genetically you're programmed to that diet. You've got your grandparents and your great-grandparents' genes. What did they do? Did they grind up their millet, or, you know, what did they eat? And that's what you need to get back to.
I'd be in trouble. I probably ate a lot of meat.
It doesn't matter. It doesn't matter. And then we develop these intolerances. The problem is we develop all these intolerances because we've been exposed by minute quantities of glyphosates and organophosphates and PCBs and every bloody thing. We've got wax around the damn apple so it shines, okay? So, sorry?
Sorry. How do you even know it's fibromyalgia? Like, how do you know that it exists?
Fibromyalgia, there's a certain classification of fibromyalgia, there's certain disease or symptomology that is associated with a condition called fibromyalgia, okay? So in relation to diet, I think it's very, very important not to get caught up in every specific diet. If you're subscribing to a diet, you might find that it works for you and stick with it, that's fine. But if it doesn't work for you and you're finding that one, a symptom might aggravate in relation to another, then you might find that you need to go back to basics. Back to basics is keeping life very, very simple. And we're limiting the varieties of foods that we used to have is much, much more, or our grandparents used to have much, much more than the variety we have now. We only have about a dozen vegetables in our supermarkets, right? And there were many, many more. Okay, so be aware that those are the ways to look at it. Let's move on. In light of neurological disturbances, we also know that we need to identify, copper, ceruloplasmin. Copper's a biggie because copper, elevations in copper. Now, in this case, you can see this copper is sufficient. That's not a problem. Ceruloplasmin is high normal, but the free copper is as dangerous as mercury to the body. So high copper levels in the body is associated, not only we know that histamine needs copper for metabolism, but it's associated directly with mental health conditions, okay? So again, we won't go into the intricacies of it. All I want to do is make you aware that there are many, many ways to identify the metabolic blocks under which our neurological system gets affected. Methylation. Has anybody heard the term methylation? Okay. Right, okay. Very simply, we'd be dead without methylation. One of my lecturers once mentioned that you're dead, you take your body, you burn it down, all you are a bunch of carbon. You compress it, you can make yourself a nice little diamond out of it, okay? So that is, we are carbon. So much for the carbon loading and all that stuff. We are carbon. Methylation involves a carbon and 3 hydrogen. So what's the idea of it all? It's to regulate homocysteine. Homocysteine, too high, inflammation, cardiovascular disease, diabetes, cancer, chronic inflammatory conditions. Too low, we can't make energy, glutathione. So when you're understanding methylation, all we need to do is get an understanding of what drives homocysteine. There's the folate pathway, and folic acid comes from foliage, the green stuff that we eat, right? That's folic acid, and that's what we need. We need enough of the folic acid to make this whole thing work. And I'm sure you may have heard of the term MTHFR, methylene tetrahydrofolate reductase. It's an enzyme, it's a gene marker. If you are homozygous for one of the genes, C677T gene, big problems. There's a lot of methylation blockages. This person is going to take a long while to heal. Some people can take a vitamin, they're jumping out of the skin. Others take a vitamin, they feel, oh my God, this neurologically upsets them, right? I can't take it, I'm getting the shakes. Others take the vitamin, they take a whole bottle, and they feel, oh, I'm not feeling a damn thing. Why? Methylation defects, okay?
Was it too high? Sorry, I missed that. Too high.
What's that?
You said too high.
High homocysteine is associated with inflammation, any inflammatory condition, whether it be diabetes or cancer, or it doesn't matter what it is, any brand name you'd like. Now, the other pathway that also drives this is the SAMe pathway. Now, without the SAMe, We cannot make our neurotransmitters. We cannot make muscle. We cannot make the heart work. We cannot make the gut work. We cannot do anything. So this is why methylation is so important. This is just going into the nitty-gritties of methylation, okay? Not going to go into the details of it all, but I'm just bringing this to your attention that your average doctor will not know about this. And if you know that these are the imbalances, the metabolic blocks in your clinical picture, you have to address that. And this is identifiable quite easily. You can identify it through genetics. So you can send a spit sample off and you can actually get your genetics. So this is exactly the same process, but except see how here we can see MTHFR driving the folic cycle. You can see methylene synthase, MTRR, driving homocysteine to methionine and homocysteine to tetrahydrofolate. So that's what we are seeing when we are looking at these markers here. Okay? Again, it sounds like a lot of gobbledygook, but all I want you to be aware of is that by using your genetics, you will be able to identify the propensity of degradation of the body or the healing capacity of the body. Because you can't change your genes, you're born with that. But you can change its expression. And by changing the expression, by putting the right nutrient cofactors, you're alleviating a lot of that inflammatory cascade. So in this particular instance, what's red is MTHFR. There's one or more mutations found, and that's where you have to focus on, okay? And then there's only one green one, which you leave behind, but then these others also these are driven by COMT, catechol methyltransferase, and methylene reductase, etc. These are all the enzymes that are involved in that whole pathway.
When you say one or more mutations, do you mean just homozygous or heterozygous as well?
Or compound heterozygous, all three of those options. And they give you an indication of the degree of methylation defect. So for example, if you are homozygous for C677T gene in MTHFR, then you have lost about 90% of your methylation capacity. Okay? And this is a little bit more information about those very gene markers. So we won't go through that. Now, the other thing that is also valuable is we know that, fibromyalgia, chronic fatigue patients, MND patients, MS patients, they all have neuromuscular disruptions. Their muscles, myelin sheath production breaks down, their nervous system breaks down. What is muscle? What is your genes? When we talk about these genes, they're only patterns of amino acids. What's muscle? Muscle is made up of amino acids. Where does that come from? It comes from the protein that you're taking in. So you can measure the amount of amino acids in your body either through urine or through blood. And again, each of these areas, this is my next book that I'm due to publish in the next couple of months, is on amino acids.
What's this test called, just amino acids?
Amino acids, these are your building blocks of protein. Why is it so important? Because it gives you an indication of amino acids are the precursors to neurotransmitters. They are important for liver detoxification. They're important for the heart to function. They're important for every pathway, all right? So it gives you a lot of information. It's needed for muscle, collagen, hormone production, all of those things.
Is how you're feeling at a particular time affect the results, or are they pretty steady?
Generally not, no. Unless yesterday you had a very high protein meal and you're taking your urine sample.
I know if you're having a good day versus a bad day.
Yeah, generally not, no. It wouldn't affect the amount of amino acids significantly enough, right? There may be a 5% up or down, but generally not, no. Right. So when we're looking at issues like, we might the level of interpretation of amino acids is so profound now that we can actually suspect, just as far as gastrointestinal health only is concerned, if you've got a low leucine, isoleucine, valine, we might suspect a digestive proteolysis deficiency or else leaky gut. Or we might suspect fat maldigestion if we've got taurine and glycine imbalances. Or we might suspect liver detoxification if we've got low glycine, glutamine, and aspartic acid. So this is the level of which we can go through it in much more detail, but for the context of this talk, I just want you to be aware that these are additional areas that you can dig deeper into amino acids, which can be done in a dried urine strip, a urine sample, a 24-hour spot, or a plasma. Now, we make energy. We know that, right? They get stored in the mitochondria. Mitochondria are little organelles. There are about 300 to 600 mitochondria in every cell. So our plants need sun, need nutrients from the soil, it needs water to make lovely pumpkins, green stuff, trees, flowers, etc. Why are we eating fats, carbs, and proteins and water? We don't make pumpkins. We make ATP, which is our energy, right? So we can actually measure the level of ATP through what we call a Krebs cycle or organic acid test. So we measure the Krebs cycle, which consists of 3 major stages. The first stage is actually what goes from the gut to the time it enters the mitochondria. In other words, here it is absorbed, metabolized, digested, excreted. Whatever's absorbed goes into the bloodstream. It's detoxified. Now it's finished the detoxification. Now it's got to translate to energy. Where does it go? It goes to the bum cell or the hair cell or the heart cell, and then it converts into the mitochondria. From this point onwards, the citric acid cycle happens in the mitochondria. And in the mitochondria, as you can see, you said, where do these amino acids come from? They're so important. There they are. They're very, very important for every step of the citric acid cycle. But we also need other nutrients. So the citric acid cycle will give us the energy utilization, the B group sufficiency, coenzyme Q10 sufficiency, gives us the balance of the happy and the sad brain chemicals as well. It gives us whether we are absorbing, metabolizing, and digesting our fats, carbs, and proteins, the ratio of pyruvate to lactate. Some people say, I feel so tired, my muscles feel so sore all the time. It's like an elite athlete running and they're going through what we call lactic acidosis, their muscles start to shake. Chronic fatigue patients, they go through the same thing, but they didn't do any exercise for it, they're just going through it. Why? Because they're developing this high level of lactate. Why? Because there's not enough of those nutrients to convert that pyruvate so that it can convert to energy. Instead, it's just sitting there causing a little bit of havoc. Okay? So you might be eating the right things, but if it's not converting to energy, we've got problems. Then the oxidative phosphorylation pathway involves, it's like a battery pack, Energy is going from one part to another to actually produce your ATP, all right? So this is, it measures a lot. One test can do that, and it's worth knowing about. And so we can go into a lot of detail, but that's what a typical test result might look like. So it identifies your B group sufficiencies, your methylation capacity as well. Methylmalonic acid will give you an indication of your B12 utilization sufficiency. So you might have your B12 levels done, and your doctor says, your B12 levels are really, really high, and your doctor says, well, you're tired, let's give you a B12 injection. Right? Now, your serum active B12 might be high. But is that translating to energy? May not. So utilization of that B12 is measured through something like methylmalonate. Okay? It's just bringing a couple of these things into play for you. So then we think about, well, okay, so now we know about, that energy production and nutrient production. But we now know that there's a huge amount of study and evidence surrounding infections. We now know that most people who have mold infestations and Lyme disease and Borrelia infestations, these are people who are actually having major chronic inflammatory conditions. We don't know what they are, but they definitely have these. So they can call them SIRS, you can call them Lyme disease, you can call them, MS, you can call it anything you like. There are many, many conditions out there, and they each have their own etiology and physiology and labs and symptomology. So we now know that Streptococcus is associated with PANS and PANDAS. We know that Lyme is associated with chronic fatigue. And this is quite a reasonably good article where you can actually have a look at some of the emerging infectious diseases associated with these chronic inflammatory conditions, which can derive from microbial factors, from human factors, whether it be environmental, and they all bugger up the microbial function in the gut. Okay? So what can you do? Generally, I would look at your HLA. When you're looking at this area, it's really good to get an umbrella view because you don't know whether you've got mold or Lyme or dinoflagellate or parasitic infestation. You don't know what it is. So the HLA-DQ studies are the best way to go about it. And if that is positive for certain areas, then you go down deeper and look at mold, or you can look at a sample. You might be living in an environment where there's mold happening there, and ever since you've moved into that house, you've become sick. So you might want to take a piece of that mold and send it off to the lab. So that's what we call an ERMI score. You might want to also look at specific molds. What are the molds in my body? So that could be that one there. Or you might want to say, look, I've got respiratory issues, I've got sinusitis, I've got all these issues, migraines all the time. There may be a bacterial or fungal issue happening in the nose, so it's a nasal swab. You might find that, look, I need to know if this test showed me that I've got Lyme, so we might need to do a Lyme serology. That's your Borrelia species, your Bacteria species, and so forth. So the HLA-DQ studies are basically, HLA is human leukocyte antigen. DQ is associated predominantly with your gluten intolerance or gluten sensitivity. The combination of DQ and DR is studied very extensively by Dr. Richie Schumacher in his work. And a typical test result will give you an indication of what your propensity would be like. For example, if you've got HLA-DQ haplotypes expressed or alleles expressed as 11 and 15 and DQ as, that's DR, and DQ at 6 and 7 and 1 and 5, you match them all up. You don't have to do that. We do that for you. But this person has 15 for the DR and 6 for the DQ. This person has Borrelia post-Lyme. That means that they have had exposure to Lyme disease. They're suffering with the exposure of Lyme disease in the past. They've been bitten by a tick somewhere along the line. Multiple antibiotic resistant staph, MARCONs. So here is where we would actually do a Lyme test, and we would also look at their nasal swab and have a look which bacterial bug caused it, which fungal bug caused this, and can we fix that, okay? So this is what they call a Rosetta Stone. Dr. Richie Schumacher again, Biotoxin Journey. It's all based on that sort of thing. Now, when you know that the patient has mold, so what do? You go and do a specific bunch of tests afterwards, once you've established that. So you have a look at their markers for mold, and there's a specific protocol involved. Now, this is a great resource. Go and have a look at Biotoxin Journey by Dr. Schumacher. If this is an area you're interested in, you can spend many, many, many thousands of hours looking at his work and his study. So that's a good thing for you to think about. Essentially, it'll affect appetite, it'll affect all inflammatory cascading, okay? It'll affect gluten sensitivity, it'll affect your blood pressure, it'll affect your hormones, and so forth and so on, your adrenals, and it goes on and on, okay? Also identify specific, as I said to you, you can take a bit of the dust or the mold, and you can go and check that out, and then you can actually measure that as well. So there's your environmental moldy risk, or you can actually look at mycotoxin assessments as well. And there's some molds that are really prevalent, things like aspergillus, and there's a number of them that are fairly prevalent out there.
You die of fibromyalgia?
Everybody dies, but can you die of fibromyalgia? You will die of any form of degradation of the nervous system as well as the inflammatory condition. Yes, you can, but everybody dies. Now, it's not an issue of, it's a question that is, I can't answer, laterally, because I know nothing about you. Okay? Yeah. All right. So, in relation to mycotoxins, I mean, these are the things that you can actually measure, all right? So in terms of all these toxins that are out there. Lyme disease is such a huge issue out there. And so Lyme is a tick-borne illness. It's typical in tropical areas, but we now note that it is, the prevalence is much higher. The various species of Borrelia and Burgdorferi are associated with Australian ones are measured here. Before they were only confined to the American species, now they are confined to the Australian species as well. So what happens is you get this butterfly skin rash, and that's a really good telltale sign. And so, it's just basically a tick. And people get chronic fatigue, they get all sorts of other inflammatory conditions, all right? And so a typical result might look a little bit like that, and it might look a little bit confusing, but at the end of the day, it's based on the amount of bands that are present. So you've got your Babesias, Borrelias, Garcinias, and so forth and so on. And then there's co-infections, such as Ehrlichias and Bartonellas as well, that are measured, okay? And so based on the bands that are present, it will give you an indication of whether the patient is Lyme positive or Lyme negative. This is merely just to give you an insight into them. I'm not digging deep into them, because each of these tests I spend an hour talking about, okay? But I'm just bringing all of these concepts together so that it opens up your vision about how you can identify the underlying causes behind what you're experiencing. So a lot of people have issues with, well, we know that we are adulterating our foods, but perhaps you lived in an area where you were in the mining industry as a child, or you are working as a hairdresser, or you are working in an environment where you're exposed to toxic exposure. Prolonged toxic exposure has been shown to be fetally transmitted. So if a father has been working in a specific environment, perhaps making aluminium cans, for example, that child, that newborn child, will have an aluminium toxicity. It's genetically predisposed. So therefore, what you can measure is you can measure metals and minerals. You can measure that in a number of samples. You can measure in hair, in urine. The hair sample will give you an indication of what's been there for a long time. What's metabolically right now? We had this patient who actually went on a ballooning, 50 years celebration, went on a ballooning experience, very windy, got a whole whiff of whatever gases they use up there, got very sick after that, okay? So, and so fortunately we had some of her urine for another test, so we managed to measure the metals at the time. So that was really good. We had the before and the after shot, and that was quite a significant case. So mercury, we know that mercury is derived either from the food that we eat, or it could be derived from amalgams or exposure somehow or the other, so we can measure that. Toxic pollutants, so we can measure all of these things, the volatile solvents, chlorinated pesticides. These are things that we're exposed to all the time in various ways. So one test can identify your alkyl phenols, your chlorinated pesticides. So these are things that we spray on our produce. Organophosphates, another spray on the produce. Phthalates and parabens, these are found in so many products out there, whether it's a cream that you slap on or a cleaning product that you use. Bisphenols are also in your plastics. You drink your water from your water bottle. You're getting your BCB load. And then your volatile solvents. You put petrol in the car, you've got some level of volatile solvent, okay?
Just a question about fragrances and perfumes and all of those things.
Mhm.
Do they kind of fit into some of these
Yes.
categories? Yeah.
Yes, they would be part of your volatile solvents, yeah, okay? But then they do use parabens as well in there, okay? So just bringing your attention, because it's so vast, I just want to bring your attention to this one, just so that you can get an idea of what I'm talking about. Alkyl phenols they are your bisphenols. It's in your you know, when you go and get your till ticket, do you want a receipt? So it's got nonylphenols, bisphenol A on it, okay? That's why it fades. It's nicely on your hands. Water bottles, plastic dinnerware, baby bottles, your triclosan. Now, bisphenol A is one of the major endocrine disrupting chemicals causing all sorts of breast cancers and prostate cancers. And that's very well published in the literature. Triclosan is found in your toothpastes and cleaning supplies and deodorants. Non-alphenols are also found in your cling wraps and so forth. So I just want to bring your attention to one of the components so that you can get an idea of what we're talking about. So a typical result might look a little bit like this, where you can see non-alphenol is elevated. So that is associated, as you know, with food packaging, with you know, your thermal sales, tickets, and so forth, okay? Then we can also go and look at where this area is rather complex. Not many practitioners know how to interpret their cytokine panels. And this is looking at the cytokine cascade, so the imbalances of T helper 1 and T helper 2. So that's your inflammatory versus anti-inflammatory components, but that's also available. So really, in a nutshell, chronic inflammatory conditions, they are multifactorial. All of the tests that I have mentioned today, they are vast and varied, and there are many more that I have not even mentioned. But it is important for you to be aware of the options that are available to you so that you can say, right, okay, I've been to so many practitioners, I haven't identified, I believe that there may be a trigger in this area which I might need to identify, okay? And you now have the opportunity, this information is not freely available, but you now have the opportunity to be able to say, hey, I need to know a bit more about this area, okay? Which organ system needs attention? Very important. You might find that there may be a specific organ system that needs more attention than another in order to alleviate some of your symptoms. Might be working on the gut first before you do something else, or the adrenals first. A practitioner will need to understand what the presentation is, what the treatment is, what the response is, and then work out whether that pathway is the right pathway for the patient or not, and what's the underlying cause. And this is why you want to know what needs to be tested. So the testing, unfortunately, all the things that I've mentioned, are not covered by the Medicare system, unfortunately. And they are all privately funded. So it is very expensive if you say, I'll have the lot, right? And there are patients that have the lot because they can afford it. And they're getting better. And there are patients that can't have the lot. So you then work with your health practitioner and identify what is most paramount for you based on your history. What have you done? What have you tried? Where have you gone? What's happened for you? What's the response? Then we can open up the doors to all these other components. So I thank you so much. A lot of these tests, or actually all of the tests, are available at Nutripath. And if you've got any questions, you're welcome to email me. I do consult as well out of Blackburn, so you can definitely contact me through that email. And yeah, I guess let's...
Is there a way of being normal again? Like feeling normal? Like you can do things on your own without other people doing for you?
When people have chronic inflammatory conditions, once the metabolic blocks are identified and corrected, and your adrenals are optimized, then you start to feel normal. But whether you will feel normal as you were when you were well is questionable. There are people that have chronic inflammatory conditions,
They can get rid of it.
and they get to a point where they're just fine. They're perfectly fine.
I'm just curious, sorry to interrupt. My dad always says to me that, like as we just spoke, the diet is really important, but how do you know that affects the actual pain? How do you know that it doesn't harm by itself?
This is why you can test what is good for you specifically. Any other questions?
Can I ask you a question about the SIBO?
Yes.
How is that related to fructose malabsorption? I know there is a relation between SIBO and SIRS.
Yes.
But is that just different parts of the gut?
Fructose, galactose, and glucose are other breath tests available.
So I tested positive to
Fructose.
and was on FODMAP for a couple of years, which was bad because
It
gut
aggravated it,
microbiome-
yes.
Well, no, your gut microbiome isn't getting different things. You should only be on a short time. So now I had lots of fructose, and I'm fine with that being on cholestyramine. But
Yes.
I'm just wondering, so like is SIBO one part of the gut and fructose
Absorption is in the upper. They're all related, and fructose malabsorption, you might be fructose malabsorbed now, but if you rectify the immune tolerance with high levels of lactobacillus rhamnosus, for example, and this is a study that was done on peanut allergy, they found that 80% of anaphylactic peanut patients could tolerate peanuts. This is a well-published study.
Really craving peanuts, which I know peanuts, they say
Okay,
cheap,
it's,
you've
no,
got
I'm just bringing to the attention of what you think you're allergic to.
Yeah.
You're not allergic to forever and a day,
Yeah.
because you can rectify the immune system in the gut in order to tolerate those foods. Now, you may not tolerate like 3 pieces of fruit. You might be able to tolerate 1 strawberry or something without the needles, but you know. Yeah. Any other questions? Did you find this overwhelming or useful?
No, it's good.
Was useful? Because when I was putting this information together, I thought, well, it's really hard without actually showing you what exactly is available. So it sounds like a lot of material, but at the same time, you've got to have a perception of, does this fit for me?
Yeah, it is kind of overwhelming.
Of course, yes. It's a language that you're not familiar with. Yes.
And what I'm curious about is, say, case studies where someone comes presenting with whatever problem and they're taken in, what's the pathway, what's the likely outcomes for different cases, and what happens.
So it's very important to work with. The confusion lies with the conventional versus the integrative model. The conventional model, your doctor is only familiar with what's on medical director, and they're familiar with the standard conveyor belt approach. Within 5 minutes of a consultation, they'll be able to look at certain basic labs. Anything beyond that is in the too hard basket. They may be interested, but they probably haven't had the time to investigate these options. And this is why integrative health practitioners, whether they're doctors or naturopaths, etc., work in this area. And because they gone to the trouble of learning this. I go to the trouble of teaching each of these areas in a lot of detail. And our doctors learn this. And this is where they can then, you can work with doctors like that. You can even consult with me if you want to, it's up to you. But these are the practitioners that know what they're talking about because they're educated in the area. They can then guide you accordingly. Right?
Somewhere I could go to see case studies.
I mean,
People that have,
right here at NIM, at the conferences, have you heard any of the conferences here at NIM? Well, you know, right here, this is what they do. It's a research facility here. This is one of many places that,
they publish sort of case studies.
yes. Yes. Yep. And there's a fair amount of published material out there as well. So, you know, there's a lot of good material out there that gives you evidence behind these strategies. All I'm doing is talking about assessments. I'm not talking, haven't mentioned any form of treatment yet. But that's not the objective of this segment. All right, well, thank you so much. Any other questions? One more?
Is it possible, I'm not sure if Peter has a list of the tests, or did every one of the slides have what the test was?
That's correct, yes. Yeah. All right.
Thank you.
Thank you so much. Thanks a lot.